you very much.
Thank you, and thank you for the invitation to come. And actually, I've slightly changed my talk because I saw my friends Rupert and Shaquille were on the program, and I knew that they would cover a lot of interesting antibody and cell therapy stuff. So I'm just going to talk about one thing, and I'm going to do it quite slowly because it's kind of complicated, but I'm going to try and take you through the story from beginning to end. So it's about a clinical, just one clinical trial, which we have been working on for the last about 6 years to open at Great Ormond Street. So you can see my schedule there on the right, which I wasn't intending. You can see my little publication here, which I was writing during the talk. Let's see. Yeah, Grayling's really is Grayling, the Justice Secretary. I'm just going to try and open that again. Old technology mixing with new technology. So as I said, I now can't see what I'm meant to be looking at, but never mind. So I'm going to try and just talk you through this. So immunotherapy can really come in 3 different forms, which is antibodies, vaccines, and cell therapies. And Shaquille will talk quite a lot about antibodies later, so I'm going to sort of skip that. And vaccines isn't really going to be covered much today. Maybe Shaquille might talk about it, and Rupert will talk a little bit about some cell therapies in the form of transplantations later on. But I want to just show you this cartoon, which tries to explain what an antibody is and how it works. And this will be important for explaining how it fits in with cellular therapies at a later stage. So antibodies consist of, is this a pointer? Oh, there you go. 2 parts. There's a part up, so these are little molecules, tiny little things, which can be made by biotech companies, or we can make them in the lab. And they're naturally existing things, which are naturally made to fight infection. But it's possible within the lab actually to generate these in large numbers. And they all are absolutely identical in that they all have exactly the same structure here. Now this is the bit of the antibody which binds something called an antigen. You can see here on this cartoon, this is a neuroblastoma cell with a particular molecule on the cell surface. This one I've drawn as an octagon is GD2, but it could be any other antigen which is present on a neuroblastoma cell surface but isn't present on the surface of normal cells, could be targeted with antibodies. The idea is that the antibody, this part of it here, which is the recognition part, binds it, and the bit hanging off shown in yellow is a bit which then is important in setting up an immune response. So that's shown in this cartoon here, which demonstrate that you've got a killer cell which specifically binds to this part of the antibody, and in the presence of things like GM-CSF or interleukin-2, if they can be added into that environment where this interaction is happening, and these killer cells get activated. Now these killer cells could be many different types of cell, which are naturally existing and circulating in the blood, such as natural killer cells, or certain other cell types which a number of us are working on as alternatives to so-called NK cells. Okay, now why am I telling you all this? So this is the most complicated slide, and I will talk you through it slowly. So here on the left is the antibody we just described. These circulate in the blood and are produced by cells called B cells. There are another type of cell in the blood called T cells, which are important normally in fighting infection. Now, on their cell surface, they have a thing called a T cell receptor. Now, this green thing shown here is the membrane. It's the surface around the outside of a cell. And a normal T cell has this so-called receptor in which it's got something rather like an antibody here, which sort of binds specifically onto a target. And inside it, it's got a whole load of other components of this complicated molecule, the important bit of which I've shown here, drawn in orange. And this is the part which is responsible for sending signals to the cell to do things. In other words, this is a T cell. When it binds, recognizes some target it's going to kill, that engagement occurs through this, and the signal occurs through this bit shown in orange. So when it meets the target, it goes mad, it divides and makes copies of itself and produces a whole load of killing molecules. Now, the thing which we've been working on for neuroblastoma for about 6 years now, as I say, which was initiated by a grant from a neuroblastoma society, is the so-called chimeric antigen receptor. Now, the concept of chimeric means it's got components of two different things. So what we actually do here is combine an antibody with a T-cell receptor. Now, you can see that we've got this sort of brick shape and this pale shape. These are the important recognition parts of an antibody. So it's possible to do genetic engineering in order to create a new type of molecule which can perform the functions in a T-cell normally given by antibodies. So I'll come on later to show you how that works in some more cartoons. Now, there are actually different flavors of these ones, and they get very complicated, but in simple terms, there's so-called first and second generations of these types of molecules. The first generation ones were used initially about 5 to 10 years ago in clinical studies, and the second generation ones have come onto the scene in the last 1 to 5 years and are much more potent than the initial ones, and that must be borne in mind when we look at the results from clinical trials. Now, I want to introduce the concept of adoptive immunotherapy or cell therapy. Now, the concept here is that it's possible to take a blood sample from a patient, perform a manipulation in the laboratory of that blood sample of the cells within it, and then select the ones which you want and inject them back into the patient. Of course, these cells which you select, shown in green, are the ones which have a particular reactivity against a tumour as a result of those receptors on the cell surface. So this is the so-called adoptive cell immunotherapy. This has been around for many years, and some of the most exciting results in cancer actually came from the North American groups such as Seattle and the National Cancer Institute, who grew out T cells which recognized melanoma from melanoma patients and injected them back into those patients, and got some very dramatic responses when this was done. Now, this is an example of one of the scans from one such patient, showing the principle that even in patients with very resistant disease or relapsed disease, it's possible to get some control of a cancer and even a shrinkage here of this liver metastasis. This is a CT scan, and this shows a liver metastasis getting smaller over, as you can see, a very long time period. So the single injection of T cells can stay around for months or years and can cause these ongoing, durable clinical responses. Now, this is I won't go through the details, but this is a summary of a lot of clinical studies done at the National Cancer Institute, actually, showing that when they introduced chemotherapy, in addition to giving T cells, was a much better clinical response. And this is all results from patients, adult patients with melanoma. But it shows a very important principle that when you inject T cells into a patient, if you give chemotherapy beforehand to reduce the number of the patient's own T cells, it enhances the survival of the T cells you give back and improves the clinical outcome. So that's an important principle. Now, this is a little cartoon of a virus. Is it possible to switch the lights down at all? For later on, it might come in handy. This is a cartoon of a virus, and we use viruses as delivery tools in order to get things into cells. It doesn't matter, Mark, if you can't. Now, in the middle of a virus is this curly thing. This is DNA. So DNA is the stuff which gives the information to cells. And a virus is therefore a method of putting new DNA into cells and changing them. So it's one of the techniques or the tools which is available for so-called genetic modification. And that becomes important, as shown in this cartoon here. Now, a virus normally has a whole load of genes inside the DNA, shown in these different colours, which perform different functions. The blue one and the yellow one in this picture are responsible for the infection. In other words, the virus makes another copy of itself, kills a cell and goes on. So those are the things which create all the danger of viruses. But it's actually possible to remove all these genes from viruses which are responsible for viral infection and to replace them with a gene which you want to put into a cell, shown here in red. So this is the gene we want to put into a cell. This is how genetic engineering of T cells works. So the idea is that you have a patient's own T cell here in the laboratory. You infect it with a virus in which you've made completely safe by removing the blue and the yellow, but you put in a new gene which encodes the molecule you want to put into those T cells. So of course the molecule we want to put in is the chimeric antigen receptor, or the CAR, and that will make the T cells reactive now against neuroblastoma. One of the advantages of doing this is that when you infect a T cell with a virus, the gene, which is now shown in blue, so this is now our chimeric antigen receptor gene, can stimulate the T cell to divide, and when it does divide, you get two new cells, each one of which has got the new gene in. So it's a way of then expanding the population of cells which have this specificity for the tumour you want to target. So now we can adapt the protocol I showed initially, and when we take our blood sample and get our T cells from it, we now add the virus at this stage. So here's the virus going in. So these become virally infected cells, but they're not infectious, they're not dangerous, but they do have the new gene in them. So when these get injected back into the patients, then now the T cells have the ability specifically to kill the neuroblastoma. And this is really showing the same thing in a different cartoon, in case you're struggling now with the concept. Here's our chimeric antigen receptor. This bit here is derived from the antibody. So this bit has been derived from an anti-GD2 antibody. So this part of the molecule recognizes GD2. This is inside, on the surface of the T cell, following our genetic modification. Here's a bit which gives the T cell receptor signaling. And here's our T cell with the CAR on its surface following the genetic manipulation. It meets the tumour. This is now GD2 on the surface of a tumour. And there's a sort of lock and key principle here, which means that when the T cell meets the tumour, the key fits in the lock, the signal gets given to the T cell, and it goes on and makes copies of itself and kills the tumour. Now, why are we using T cells rather than antibodies? Well, T cells have a number of potential, striking advantages over antibodies. One is that they actually have a natural mechanism to get into tumours. They're able to get out of blood vessels and into tumour and infiltrate, especially if you create a sort of lots of danger signals within the side of the tumour. They have this ability to recognise tumours as foreign. They kill directly. They have lots of very potent killing mechanisms. Picture here of this electron micrograph showing a T cell bouncing onto the surface of a tumour cell. And they can release different things. So we can put whatever we want to into the T cell, and then it'll discharge it in the region of the tumour. And it sets up an inflammation which can recruit other cells. Now, this technology has been applied most successfully in the last few years in leukemia. And in the same way that neuroblastoma has GD2 on the surface, which is pretty specific for neuroblastoma cells, leukemias of a particular type have something called CD19 on their surface. This is showing an example from University of Pennsylvania of a trial in which this is a blood sample being analysed on a flow cytometer, and this is a heat map. So this basically every dot represents a cell. So there's around about 5 million cells here in this little blood sample, all of which are leukemia cells. When a blood sample taken 1 month later after giving this infusion of chimeric antigen receptor gene modified T cells results in the elimination of this. So, is this a simple and safe procedure, or is this complex? So I'll go on to describe a little bit about how we're now applying it in neuroblastoma. So as I mentioned, this study, which I hope will open around about April-ish, at the earliest of next year, is funded by Cancer Research UK and the Drug Development Office, which is a very big team. And we have a team of investigators at Great Ormond Street and UCL. And I need to point out my co-investigators, Karen Stratoff and Martin Poole, who are the co-investigators on all my grants, which have been doing this work. So Martin, when he worked at Texas Children's Hospital, did the first clinical trial of anti-GD2 chimeric antigen receptors for neuroblastoma. This was quite a small trial initially. It was a phase 1 study, and importantly, it used this first generation chimeric antigen receptor, so the one which hadn't yet achieve a full optimization. And also these patients didn't receive any lymphodepleting chemotherapy, so they didn't get chemotherapy before the T cells, which we now know would have been an advantage, but it wasn't known at the time. So out of the patients on that trial, there were a number of clinical responses, and this is one from the publication showing an MIBG scan with the disappearance of this metastasis over a period of about 4 months in this case. And this is the data I showed from the CD19 study. But as I said, there are some possible dangers with this. If we're genetically modifying T cells so that they now see the tumour and go mad, make copies of themselves and release lots of chemicals at the tumour site, will it have any toxicity? So I'm going to attempt to show a movie, and I'm going to switch the volume up on this and stick the microphone down and see if it works. And if it doesn't work, I won't show it.
Last February, Emily Whitehead was a very sick 7-year-old. She had leukemia. Treatment had failed twice, and the cancer had returned. Her parents brought her here to the Philadelphia Children's Hospital, where doctors were trying a new, untested treatment.
We were down to 48 hours of making a decision, or she could start to have organ failure, and so we didn't sleep for a couple days, and we ended up here.
The treatment meant taking cells from Emily's immune system, so-called T cells that normally kill cancer cells, but in her case just didn't recognize them. Doctors genetically engineered the T cells so they could go after cancer cells. It worked, but too well. The beefed up T cells made Emily's immune system overactive. She became critically ill.
They took us out in the hall and said, there is no room to get sicker. She is as sick as you can get. You know, they said, you should call your family in. There's a pretty good chance she won't be here in the morning.
Happily, Emily recovered. Doctors managed to control the unexpected side effect with drugs.
We checked her 3 months and now 6 months out from her treatment. She has no disease whatsoever. She's going to school. She tells me that she feels perfectly fine. So she really has benefited very significantly from the T-cell treatment.
Like Emily, 9 out of 12 patients in the trial benefited. Most children with her kind of leukemia respond to standard chemotherapy, but for the 1 in 6 who don't, this still experimental treatment offers renewed hope. Lawrence McGinty, ITV News.
Okay. So that's just from earlier this year, and I've wanted to show it for 2 reasons really. One, because of the potential toxicity of this treatment, which in this child's case was very marked, and the actual approach and the actual virus which they used in that study is pretty much identical to the anti-GD2 second generation chimeric receptor, which we will use in the trial as well. So our expectation is that we may see some responses, but they could be very dramatic responses, and children may need to be in a very safe environment. And for that reason, we have gone to some lengths to add some additional safety features to the receptor or the gene therapy which we will use. So there's been a lot of optimization of our receptor, and as I said, it's essentially the same structure as the one used in Children's Hospital Philadelphia for leukemia. And this is showing in the laboratory. These are T cells actually here, which in the presence of GD2 go mad. So this is like a single T cell here, and this is after a week in the presence of GD2 they start dividing, so making like 1,000 copies of themselves. So that's potentially quite dangerous, as you can imagine. This graph shows the amount of IL-2 produced by the cells in the presence of GD2, which is not seen in its absence. So we felt we needed to put in something called a suicide gene as well. So this is a molecular trick. So essentially the virus, as well as producing the chimeric antigen receptor, produces a so-called suicide gene, which creates this thing on the cell surface next to the chimeric antigen receptor. And it's so designed that if we add an antibody called rituximab to the patient, it will bind this and selectively destroy all the cells which have been genetically modified. So we hope not to have to use this suicide mechanism, but it is potentially there available if needed. And this is the cartoon of the study and how it will work. So we've developed a technique to use old G-Csf mobilized apheresis. So those of you who've got children who've gone through high-risk neuroblastoma treatment will know that they've undergone a G-Csf mobilization and had these cells stored. So most children now have additional bags left over after the high-dose treatment, which are available for other purposes in the event of relapse. And the trial will be for relapse. So we can wash these cells and in a particular way, and we find we can get quite healthy cells out, which we can infect with a virus, which is contained in these bags, then do a selection process and freeze them down and check that they work okay. And if they do, then a child who's eligible could undergo the chemotherapy to deplete their own T cells and have their T cells injected back. So this will be a phase 1 dose escalation study. So there will be 3 to 4 cohorts of patients, so between 9 and 15 patients. We're going to just give a fixed number of T cells. And we're going to start in just one centre, our own at Great Ormond Street, with a view to opening it in other centres if it's successful. So that was all this is just showing the progress timeline. As you can see, it started in 2006, and now here we are, December 2013. Next week, the clinical vector will be delivered from Germany, and we can do the final scale-ups prior to the regulatory application. Now that's where things could be delayed. They may or may not be delayed at that stage, depending on what the regulators say. Now, just my final slide. I was asked to give an overview of the other stuff we do. So as well as doing this trial, we do quite a lot of basic research in neuroblastoma in the lab. And what we're particularly interested in is finding new antibodies to target different things on the cell surface. So we have a big program looking for ALK and anti-ALK antibodies, and Shaquille is something of an expert at B7H3, but it's another target which we're interested in and have made some antibodies against. So we wish to put these into chimeric antigen receptors to test. And this work with ALK we're doing in collaboration with Andy Pearson and Lou, who's here on the front row. And the other big area of work which I do is using gamma delta T cells, and Rupert is an enthusiast for these cells as well, and we'll tell you a little bit about why they might be important in haploidentical transplants in the afternoon. So that was, and then finally another area which I think is very exciting, may or may not be the answer, is so-called bispecific antibodies. Rupert may or may not say something about those later, not he says, but this is an area which hasn't yet been developed in neuroblastoma with GD2, but I think would be an interesting area to explore. So thank you for your attention, and be delighted to talk to people at lunchtime. I have to leave sort of mid-afternoon, or I shall be in big trouble at home, so if anybody wishes to have a chat lunchtime, that would be great. Okay, thank you.